Hi everyone, welcome to our new lecture. This is our first post-midterm lecture, so I just want to say to all of you who made it this far, congratulations. Looking at the mark for the midterms, I think it went pretty well. Most of you guys got pretty good marks for both the labs and the lecture midterms. The memos will be available this week, so that should, if you have any questions or whatever, feel free to address them to either myself or Kieran if it's maybe from the lab exam, and we'll happily explain anything. Just a reminder, we are now done with the first half of the course, so you won't be tested on that material again. The final exam will also be non-cumulative, so it'll only the content for it will be the back half of the lectures and the back half of the lab content, so you won't have to restudy what you just studied for the midterms. Okay, so that little bit of admin out the way. Today we're going to be talking about classification, phylogeny, taxonomy, and nomenclature. So just before we get started on this, this little picture we have here of the eat, survive, reproduce, eat, survive, reproduce, eat, survive, reproduce, eat, survive, reproduce, and then what is it all about? It's a pretty funny picture just to get us into today's lecture, but I would like to recommend, if you guys haven't done so already, I would recommend you read the book, The Selfish Gene, written by Professor Richard Dawkins, who's an evolutionary biologist, world-renowned evolutionary biologist. And in that, he addresses what the meaning of life is from a biological and a genetics perspective, which is very, very interesting and a very, very good read. If you're interested in going into the biological sciences, I strongly recommend you read the book. I won't test you on it or anything. It's just kind of for your own benefit. Okay, so moving on. Looking at taxonomy. So what is taxonomy? So taxonomy and nomenclature. Taxonomy is the branch of science concerned with classification, especially of organisms, also called systematics. And taxonomy is a hierarchical organizational system that groups organisms based on trait similarity and evolutionary history. So this would get you to naturally related groups of bacteria. You can assign order to minimize the chaos. We just group these organisms together just for our own understanding and to improve our conceptual understanding of where these organisms came from and what organisms are more closely related to which other ones. And it's also a means of identification. So then nomenclature, which is a scientific name, which is given to organisms because it provides commonality across all languages and all regions. It provides standardization. So regardless of where in the world you are, if you use the scientific name for an organism, that remains consistent. So it just makes it easier to communicate about these organisms. And then Linnaeus, which is the standard naming system, which was based on Latin. And the reason it's based on Latin is because most scholars at the time, the 18th century, spoke Latin. Thus, it became the go-to language to use. And it's a binomial nomenclature. I'm sure you're all aware of this at this point, where we refer to organisms using both the genus and the species monikers. So some major dates in taxonomy, and to show that taxonomic classification itself has sort of evolved or changed or improved over time. So, in 1735, we had two kingdoms. We had the plant and animal kingdoms. And then in 1857, bacteria and fungi were added, but they were put into the plant kingdom. Then in 1866, we had a new kingdom for protists, which was supposed to include bacteria, protozoa, algae, and fungi. And then in 1837, the prokaryotes were introduced for cells without a nucleus. And that definition was then changed in the 1961s to include cells in which nucleoplasm is not surrounded by a nuclear membrane. In 1959, we saw the introduction of the kingdom of fungi. In 1868, the kingdom prokaryotes was proposed. And then in 1978, two types of prokaryotic cells were found. So just to give you a bit of a more background here, once upon a time, as we just saw, all living things were classified into two kingdoms, namely plants and animals, where animals included every living thing that moved, ate, and grew to a certain size and then stopped growing. Conversely, plants included every living thing that did not move or eat and that continued to grow throughout its life. However, after some time, it became difficult to group some living things into one or the other. So early in the past century, the two kingdoms were expanded into 5 kingdoms. That's what we see over here, the 5 kingdom systems, where we had Protista for your single-celled eukaryotes, fungi for fungus and related organisms, Plantae for plants, Animalia for animals, and Monera, these were your prokaryotes. Many biologists now recognize 6 kingdoms where they divide the Monera into eubacteria and archaeobacteria, just as a bit of a more detail there. Then you'll see this system is based on your phenotypic trait. So what is a phenotypic trait? Your phenotypic trait, or your phenotype, is the term that is used in genetics for the composite observable characteristics or traits of an organism. Very key there that you're aware that it is the observable trait. So it's what you can actually see. This isn't down to genetic differences within the cell. This is physical differences that you can actually observe. The term covers the organism's morphology or physical form and structure, its developmental processes, its biochemical and physiological properties, its behavior, and the products of behavior. Notice how it includes your biochemical properties. So tests in biology in microbiology that test for that would also be used to divide organisms into different kingdoms. It counts also as physical traits. Now we have our 3-domain system, which you're all familiar with at this point, your eukarya, your bacteria, and your archaea. And then these kingdoms are then divided into categories called phyla, and each phyla is divided into classes, each class into orders, each order into families, each family into genera, and each genus into its species. A species represents one type of organism, such as a dog, tiger, or shark. So if you look at the common amoeba, it would be amoeba proteus, again with that double, the genus and species moniker. Note that species names should technically be underlined or written in italics. It's more common to see it written in italics though currently. Okay, so looking at our organisms, or the 3 domains, here are some defining characteristics that you can use to separate. So if you look at cell type, you have prokaryotes, and eukaryotes. And then you can look at the cell wall composition of these cells to divide them into different groups. So if it varies in composition, but it does not contain peptidoglycan, that is a characteristic of archaea. If it does contain peptidoglycan, it belongs to bacteria. And again, if it varies in composition but contains carbohydrates, then it is eukarya. So you can go through all of these and get these characteristics from them. I mean, antibiotics sensitivity is another interesting one, where archaea and eukarya are not sensitive to antibiotics, but our bacteria are. So just some of the defining characteristics of these organisms. So where does this 3-domain system come from, and what does it really tell us? So organisms are typically classified by cell type into this 3-domain system. In addition to differences in rRNA, the 3 domains differ in membrane, lipid structure, transfer RNA molecules, and sensitivity to antibiotics, as we just saw. In this widely accepted scheme, animals, plants, and fungi are kingdoms in the domain of eukarya. So you can see within eukarya, you have all those kingdoms. Whereas the domain bacteria includes all pathogenic prokaryotes, as well as many non-pathogenic prokaryotes found in soil and water. Photoautotrophic prokaryotes are also in this domain. Then the domain archaea includes prokaryotes that do not have peptidoglycan in their cell walls, and they often live in extreme environments and carry out unusual metabolic processes. Remember our extremophiles, we've discussed some of these before. Some major types of archaea would include your methanogens, your extreme halophiles, and your hyperthermophiles, which you can see over there. So something that I do want to point out here, though, is notice how there's these lines where there's mitochondria here, and then they're drawn a line to eukarya, and then there's chloroplasts here that's also then been drawn to eukarya. So what does that mean? What is that? So that points to what is referred to as the endosymbiotic theory, which is an evolutionary theory that explains the origin of eukaryotic cells from prokaryotes. It states that several key organelles of eukaryotes originated as a symbiosis between separate single-celled organisms. So, yeah, here we go. Regarding the endosymbiotic theory, there's your full definition for it. So, what is our evidence here? Our evidence that you can see here is the organelles that we are referring to, specifically such as your mitochondria, has certain characteristics that just make us think that it might have a prokaryotic origin. So if you look at the ribosomes in a prokaryotic cell, it's 70S. If you look at a eukaryotic cell, it's typically 80S. Yet the eukaryotic organelles, specifically mitochondria and chloroplasts, also have the 70S ribosome. And then how do these cells typically grow? Prokaryotes typically by binary fission, and eukaryotes typically by mitosis. But these organelles again grow by binary fission. This just and one really strong piece of evidence is also the DNA. So prokaryotic DNA is circular, eukaryotes typically linear, and then these organelles again circular. So essentially, what this gives us the idea is that these organelles were way back when, they might have been completely independent organisms that were absorbed into eukaryotic cells and then transferred their function and still retain their functionality, and thus they evolved to fulfill the function they are fulfilling today. And that is why they still have these similarities to prokaryotic cells where the rest of eukaryotic cells do not. So in a nutshell, that is what endosymbiotic theory is. It's really pretty interesting, guys. Okay, so now we move on to phylogenetics. So what is phylogenetics? So that is the study of evolutionary history, also known as phylogeny, and the relationships among individuals or groups of organisms. These relationships are discovered through phylogenetic interference method that evaluate DNA sequences. So that is what this would look like. That is our phylogenetic tree of life with our 3 domains and their various kingdoms inside of them. And this tells us, the phylogenetics tells us which organisms are more closely related, where do they share an ancestor. I would like you guys to recall back to our LUCA, our last universal common ancestor, which would be somewhere down there, which was not the first life on Earth. It is it is thought to be the first, the earliest life form that still has surviving ancestors today. That is what LUCA is. Just as a note here, that in a phylogenetic hierarchy, grouping of organisms according to common properties would imply that a group of organisms evolved from a common ancestor. Again, LUCA down here, and all these branches would actually have, I'll be going into some more details. I don't want to do too much now, but they all stem from a single universal organism. Think of it as, it's not that animals move and then become fungi, that they down here they shared some universal ancestor and then evolution drove them into different directions. And then because they have this universal common ancestor, each species would retain some of the characteristics of this ancestor. Okay, and one major portion of molecular phylogenetics is the use of ribosomal RNA. So why is rRNA used? Well, it's one of the few gene products that is present in all cells, meaning that the genes that encode the rRNA are a sequence to identify an organism's taxonomic group, calculated related groups, and estimate rates of species divergence, because again, they're found in all cells. So as a result, many thousands of rRNA sequences are known and stored in specialized databases such as the RDP2 or SILVA. So in evolution biology, conserved sequences, you can see here they're used because they're shareable organisms and they have slow rates of evolution in this region of the gene. So this is a conserved sequence that is known as what due to this highly conserved region, but it also has a highly variable region. So what is the difference between these two? So conserved sequences are identical or similar sequences in nucleic acids. This can be DNA or RNA, but for this current purposes, we're just going to be looking at RNA or proteins across a species, or it can be within a genome or between donor and receptor taxa. And this conservation indicates that a sequence has been maintained by natural selection. A highly conserved sequence is one that has remained relatively unchanged far back up the phylogenetic tree and thus far back in geological time. Conversely, these variable regions are genes that differ among different species. So this RNA is really unique because it allows us to look at the consistent portions, the unchanging portion, if you want to see it that way, and then the one that's highly variable that would change between different species. And there you can see some of the variable regions. So how do we determine how related these organisms are? If we have our sequence now, the first thing I want you guys to do, if you're ever given a bunch of sequences and asked, which are more related and which are not, the first step is to align the sequences. So here you can see some examples of different organisms, and we have aligned their sequences. And now you move along and you note all the differences. And then typically the more differences you find, the longer it would be before you will find a common ancestor. So the less related the two organisms are. If they have few differences, they're obviously more closely related. So that is how you would end up with something like this. So if you were given all these species with their relevant genetic sequences and you were asked to draw up a phylogenetic tree, the first step would be to compare these sequences in a pairwise fashion. So if we start with A and B, you will physically go through and look for any differences between these two, right? And you will note any that you find. In this case, there are 0, just to make it really confusing. Now what I want you guys to do, the next step is you're going to draw up a table where you write down species A, B, C, D, or you can just use the A, B, C, D, E until where it goes and on the top, and then you denote the differences between the sequences. So you start then from A to B and you note that there are 0 differences. Now you would move on to A to C. So from A to C, how many differences are there? And if you do that, you will find that there are 4 differences. Thus, you will fill in the table and you will write down that there are 4 differences. You will then complete this for every sequence pair to find all the differences. And then you will use the table to identify the sequences with the fewest differences between them. These small differences, again, indicate that these organisms are very closely related and must be placed close together. So if you do that with this entire process, you will find that A has to be close to B, C to D, E to F, and G to H. So you actually start on the right-hand side. Sorry, my dogs are just going insane here. Don't know, I think someone might be at the door. You have A and B being close together, G and H, E and F, and C and D. And you sort of view this as you start on the right side and you sort of work towards the left. That's how I've always envisioned this because I think it's way easier. So once you have your initial closely related pairings, what you do then is you redraw the table, but now you used group pairs to replace the single sequences. And then you compare these using average differences between the pairs. So what would be the average difference between A and B and C, A and B and D, and so on and so forth. Once you have done this, the smallest average difference between the pairs and other sequences will inform how you will place them in the phylogenetic tree, with smaller differences being placed closer together. So that will give you your next step up. So for example here, you'll find that G and H and E and F, the average differences here are much closer than between E and F and A and B, for example. That's why it's so placed so far away. So now essentially at the end, once you've gotten all your differences and you've repeated this process until there are no more pairings to do, you redraw the table using the newly paired groups and repeat until the entire tree has been revealed. I recommend you guys to go watch this video. It's a much more in-depth breakdown of how to do this process. I just feel we're a bit pushed for time. I can't really explain it more, and it's unlikely that I'll give you a question like this, but I might. So definitely do go view this video. Something that I do wish to note, though, is that the tree itself can differ, and it could still be correct in how you arrange your branches. So to explain that, we're going to be looking at the importance of branching. So my first question in this slide is going to be one that's really important. How do you determine how closely taxa are related to one another? The answer there is that you rely on the branching pattern. That is what is telling you how closely organisms are related. It's a very common misconception that taxa that are adjacent to each other on the tips of a phylogeny are more closely related to one another than they are to taxa on the distant tips of the phylogeny. That is not correct. So in actuality, in phylogeny, information about relatedness is portrayed by the branching pattern, not by the order of taxa at the tips of the tree. Remember, you're constructing this sort of in a reverse sense where you are starting at the tips and you're working your way in. So looking at the tips will not tell you anything about how related the organisms are. You do need to look at the branching pattern. So organisms that share a more recent branching point. So if you look at here at our example, C and D have a branching point over there, where B and C have one over there, meaning C and D are much more related to each other than B and C, for example. So if you look at this example for some more detail here, taxon A is also adjacent to taxon B and more distant from C and D. See, so A is next to B and far away from C and D. So that would make you incorrectly think that A is more closely related to B than it is to C and D. And that is not true. They share a branching point up here, meaning it is equally related to A is equally related to B and to C and to D, because the last common ancestor would be up here. So the common ancestor for A to all three of these, to B, C, and D, is up here, meaning it is equally related to all three of them. So you have to rely on this branching pattern to tell you more about the relatedness of these organisms. So similarly, if you look at B then, which is next to A, B is actually more closely related to D than it is to A because of this branching here. This branching up here that it shares with the common ancestors of taxa B and D can be found up here is closer than the one with A. Thus, B is more related to D than it is to A. So very, very, very important that you guys understand how these trees work and how to interpret them. I don't want you guys to misconstrue information, and you can definitely expect to find questions such as these, because it's common misconceptions that I feel should be any biological sciences student should be able to read a phylogenetic tree and glean the important information from. So, and then we get to the bottom portion, because it's the branching that is important, not the tips of the trees, as we just discussed. All of these trees are actually the same thing. They convey the same information because they have the same branching pattern. So if you were ever asked in an exam, order these sequences, how you arrange them at the tips is not where you are, is not what matters. It's what your branching pattern says. Because you read a phylogenetic tree by looking at its branching patterns. I can't stress that enough. Okay, so as long if you understand, if you understand these misconceptions, or if you don't, I'd definitely advise you to read up more on it because it is really, really important. But yes, so all these things say the same thing because the branching patterns are the same. Okay, so how do we determine these traits for microbes? As microorganisms, they typically are not preserved in fossils. That's an advantage we have when we're working with, plants and animals. And if you look back at dinosaurs and all these wonderful things, you can look at the relatedness by looking at their characteristics using fossils. But what we have to use for microbes is we have to use a rate of change in DNA to estimate the time since a common ancestor, which is given to us because DNA mutates at a roughly clock-like rate. So essentially, if you know at what time or how frequently something is likely to change, and you know the total differences between them, the two organisms, you would be able to calculate then how much time roughly it would have taken to get from A to B if you know the differences. So if you know the genetic differences between organism A and B, and you know the rate of change, you can then calculate the time it would have taken for them to get from A to B, or to since they would have shared a common ancestor at least. Not really get from A to B, but you understand what I'm saying. So these relationships are typically discovered through phylogenetic interference methods that can evaluate observed heritable traits such as DNA sequences or morphology under a model of evolution of these traits. Generally, morphological characteristics co-vary with DNA sequences dissimilarity. So that's what we find here is you have these organisms where if you look at their characteristics, the morphology, you might find that there's a large difference between them, but if you look at them genetically, they might be more closely related, and that is more often than not caused by horizontal gene transfer, because that can cause closely related organisms to have very different phenotypes. So they will have a different physical appearance, yet their genetics will indicate that they are much more similar than you would perhaps expect. So essentially what just the take-home message here is when a phylogenetic marker DNA tree disagrees with a morphology-based tree, which is what we find here, it is typically a sign of horizontal gene transfer. So that begs the question, what is horizontal gene transfer? So essentially, a horizontal gene transfer, first of all, this diagram here, you can kind of view it also as reflecting total gene flow. How are these genes changing and adapting and improving, or being improving is not the right word. How are these genes being transferred to the next and transformed into the next generations of organisms over a very, very long time period? So with horizontal gene transfer, there are three mechanisms where genes can be transferred from one organism to another without requiring replication. You don't need the organism to go through a binary fission if it's a bacteria, for example. There are three mechanisms, and they are transformation. So in transformation, a bacterium, the recipient bacterium, will simply take up extracellular donor DNA. So there's some DNA floating around, say in water, for example, and this organism just essentially, takes it up, incorporating it then. What is transduction? So in transduction, this is where you need a bacteriophage. So the DNA from a donor is packaged in a bacteriophage, which then will infect a recipient bacterium. And thus you see that gene being transferred. So if you have one little microbe here and there's a bacteriophage that then infects it, and the replicate in lice, and the new bacteriophage then has some of the genetic material of this original bacteria. Remember, just as a disclaimer here, bacteriophages are those small little viruses. And then that bacteriophage then goes and infects another bacterium. It can take the DNA it has just taken from the first bacteria and transfer it to the new one. And then in conjugation, this is probably the one that most people think about when they think of horizontal gene transfer. This is essentially where the donor bacteria transfers DNA to their recipient by forming that sex pilus. So almost by mating, if you want to view it that way. It's not really mating, but there is contact between the two bacteria, and then the one transfers its genes to the other one. So something that I do want you guys to know here, when we are moving back now a bit to taxonomic classification, there's a useful little a rhyme that you can use to remember the order of domain, kingdom, phylum, class, order, family, genus, species. And that's dear King Philip came over for good food, for good soup. So, just as a reminder again, this tells us that your domains are divided into kingdoms. Kingdoms are divided into phylums. Your phylums are divided into classes, classes into orders, orders into families, families into genus, and genus into species. And remember, for typical nomenclature, when you write, if you look at something like Escherichia coli, you have Escherichia being the genus and coli being the species. So you have, you need that genus and species for the nomenclature. Okay. So something that I want you guys to know, some notes about the taxonomic hierarchy that you should be aware of, is that ideally each taxonomic group describes a clade or a monophyletic group, meaning that it consists of an ancestral species and all of its descendants. This is the ideal scenario, because you can then notice the distinguishing characteristics for each taxonomic group. A clade or a monophyletic group essentially just describes a branch that includes a single common ancestors and all its descendants within a cladogram. So here there's a universal common ancestor that has the ability to purr, and it gave rise to all domestic cat-like species. But then there is if you go a bit further back, there's a universal common ancestor that could retract its claws, you're giving rise to leopards and then your other cat species. So you have this kind of neat little grouping of organisms. But of course, as is often the case, there are exceptions, so that you can also have taxonomic groups that are paraphyletic. So a paraphyletic group is a monophyletic group from which one or more subsidiary clades or subsidiary monophyletic groups are excluded to form a separate group. Here, the birds are a good example. So as you can see, this is, these birds are typically excluded from reptiles, from reptilia, because they have just such unique physical features that they just simply don't share with the rest of the members. And this leads to taxonomic classification being based usually on a mix of evolutionary history and defining characteristics. And that brings us to one of the the great debates. So if you go to ornithology, so that's birds conference, they'll tell you that, as you can see, herpetology is actually a silly field. Reptiles are actually more closely related to birds and mammals than to amphibians. It should be broken up with lizards folded up into ornithology. And then, the guys on the other side of the spectrum just essentially saying that, ornithologists are actually just assholes. So it all depends on how you, group your organisms and how you view this paraphyletic, monophyletic thing. So getting back to our bacteria or our microorganisms, just to kind of move away from birds and reptiles and whatever else. If you look at something like baker's yeast, this is what our full taxonomic hierarchy would look like. It is in the domain Eukarya. It is a fungi. It's in the phylum of Ascomycota, the class Hemiascomycetes, and then its order, its family, its genus, and then its species. The same thing with if you have bacteria, for example. Note though that there are no kingdoms for archaea and bacteria. That's really important. That's something that I wanted to point out on this hierarchy. So there you go directly from your domain to your phylum. So you go bacteria, protobacteria, gamma protobacteria, enterobacterialis, enterobacteriaceae, Escherichia, and then E. coli, which is our E. coli organism. So this is a breakdown of what you can the phenotype associations for alpha proteobacteria. So something, it looks insane and it looks very confusing, but it really isn't if you just break it down into its basic components. So first of all, just like in animal phylogenetic groups, bacterial taxonomic groups can have distinguishing features. For instance, if you look at Rickettsiales, which is on this side over here, which includes the organisms responsible for typhus and Rocky Mountain spotted fever. They are an intercellular pathogen. They have a small genome size, so that's indicated by this little dot, and it's bigger dot. Small dot means small genome size, large dot means large genome size. It has adapted to live inside the host, and it has a low GC content. So it tells you about the habitat, it tells you about the GC content. So your GC content is your guanine cytosine content, and then your genome size. So if you go here, you can look all of these up, and you find that they typically have smallish genomes. They typically have a low GC content, and then their habitat is as intercellular pathogens, meaning they're usually inside of animals. Now, on the other side, you can look at something like Rhodobacteriales, which are up here. They have a larger genome size. You can see that with the larger, represented by the larger dots. They have a high GC content, gives you that blood red color over there. And they almost live exclusively in aquatic environments, shown by the blue over there. So it gives you a very nice breakdown of the habitat of the organism, its guanine cytosine content, and how large its genome size is. So, but something that I want you guys, I'm not going to provide you with the answers here, but I want you to definitely go look at this. Very important, because it's sort of to make sure you understand and test your own understanding. Which of these orders do you think are monophyletic and which are periphyletic? So just think on that. Maybe try and find the answer back home, and yeah, just make sure you understand the theory behind it. As you've seen now with my exams, I don't only test, if you can parrot work back to me. I do like to test some understanding, some insight, to make sure you guys are really grasping the work. Okay, so now moving on to how these names are given. How do you decide what the name of an organism is? Typically, that's decided by its discoverer. So if you look at something like Pasturella, these were named, well, names can either be descriptive, so Streptococcus tells you exactly everything you need to know about the organism, or it can be named after the discoverer. So Pasturella are very small, non-motile, non-spore-forming, gram-negative bacteria that are coccoid, oval, or rod-shaped. They were actually discovered by a guy called Peronchito in 1878, but they were named after Louis Pasteur, who first isolated and described the bacterium as the cause of fowl disease in 1880. And then Escherichia, you guys knew I was going to use this one, was first discovered by a guy called Theodore Escherich, and that is where the names obviously came from. Species names can be very descriptive. For example, if you look at Staphylococcus aureus, so the aureus refers to gold, the color. And then coli, from Escherichia coli, refers to colon or intestine. And then Staphylococcus epidermis, this epidermis refers to the skin. So That is how these organisms are named. The genus typically is either descriptive or it's named after its discoverer. And then it has it's either italicized or underlined, but it has an uppercase first letter, as you can see here, Staphylococcus, Staphylococcus. And the species name has a lower first letter. You can see that over there. And it's also italicized. And this essentially just modifies the generic name, provides you with more detail, if you will. Okay, so here are just a breakdown of some scientific names and where the genus name came from and where the epithet came from. So if you look at Klebsiella pneumonia, it honors Edwin Klebs, and it's the source of the epithet is from the disease pneumonia, of course. And you can just go through this table just to give you an idea of where these organisms came from. I'm sure if any of you took Latin, I actually had a year of Latin at school. Can't remember any of it, but I did have it. I think it would be very, very handy to have. I know back in the day, many science majors were actually encouraged and required to take Latin at a university level. And I think it helps a lot with the breakdown of organisms, because if you can understand what you're reading, it's essentially just telling you what the organism is and what it does. Okay, so now moving on to characterization and identification. So how do we characterize and identify these microorganisms? There are a lot of techniques to identify these organisms. You can have cultural techniques where you use different temperatures, pH, you can min-max. You can use microscopic morphological reactions, something like the Gram reaction or endospore staining. You can look at metabolism. This would be biochemical. We're still going to do some fermentation in vivo tests in the lab, so I'm not going to bore you guys with the details too much. Then you can look at the genetics. We've already seen examples of the GC ratio being used. We can also use DNA fingerprinting, which we'll be discussing a bit later on, RNA sequencing, which we'll also be touching on very, very briefly. And then phage typing. So what is phage typing? Maybe let me just move back just to make sure I have said this. G plus C ratio is essentially the percentage of nitrogenous bases in DNA or RNA molecule that are either guanine or cytosine. So phage typing is actually pretty ingenious. You can see an example of what that looks like over here. So phage typing is a method that is used for detecting single strains of bacteria. It is used to trace the source of outbreaks of infections, and it relies on the fact that viruses that infect bacteria are called bacteriophages, and some of them can only infect a single strain of bacteria. So essentially, these phages are then used to identify the different strains of bacteria within a single species. Again, this is an example of that. So how is that done? So essentially, a culture of a strain is grown in agar and dried. A grid is drawn on the base of the petri dish to mark out the different regions. And then each square is then inoculated by a different phage. And the phage drops are allowed to dry, and then the petri dish is incubated. And the susceptible phage regions will show a circular clearing. You can see those tiny, tiny dots in there. Those are the circular clearings, which essentially show that the bacteria in these regions have lysed. And that is how you can differentiate. You can use the different phages because you know what you put where, and you then tested the susceptibility, and you know which bacteria are susceptible to which virus, which phage, and that's how you can determine which bacteria this is. Pretty ingenious, actually. And some other things, you can also use serological methods using antigen and antibody reactions. Also, this is also called serotyping. And we can also do some protein and amino acid analysis. I'm not going to go into much more detail because we're already pretty much, we've already used quite a bit of time here. So again, just to break down here, you can do metabolism. You can do identification using metabolism via a dichotomous key. So what is the dichotomy? A dichotomy is essentially two choices. So if you look at something like an enteric or intestinal bacteria, the first question, can they ferment lactose? No. yes, and then you go through all the questions, and then based on the answers to these questions, you can get down to identify the organisms. Typically, this doesn't tell us the species. It can only get us down to a genus level more often than not. So you would need further testing then to differentiate between different species within a genera. But at least this gets it gets you on the right track when you are identifying an unknown microbe. So there are also some forms of rapid identification. I thought this was a pretty interesting one to explain to you guys. So essentially, this is a tube that is containing media for 15 biochemical tests that is inoculated with an unknown enteric bacterium. So essentially, if you go into the emergency room with some stomach bug, you ate something you shouldn't have, and now you are just very sick. This is something that medical professionals can use. They can take a sample, inoculate it in the tube, and then observe the results. And based on the combination of results, they can then identify the organism. That's done essentially by, the value for each positive test is circled. So if it's positive, you see there, circled, 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 circled. So you have that 4 and that 2 giving you a 6. There you go. And you have 2 giving you a 2. You have 2 and a 1, 3, 4. I don't know why that becomes a 5 and a 2 and a 1 becoming a 3. I think that's a typo. This should be a 4. And then you can read on from that. That code will then give you a code number, which is then related to a specific organism. Then also looking at genetic identification. So again, we can look at your base composition with your GC content being a frequently used one. So for example, something like actinobacteria have a very high GC content, and that can distinguish them from other groups. And then there's also DNA fingerprinting, PCR, and RNA sequencing, which we'll be discussing very briefly now. So moving directly on to DNA fingerprinting. So DNA fingerprinting, or DNA gel electrophoresis, is a technique that is used to separate and identify DNA fragments based on size. DNA fragments of various sizes are loaded into a porous gel made from agarose, which is a carbohydrate found in red algae. An electric field is then applied, and the fragments will migrate through the gel thanks to the to the negatively charged phosphate groups in the DNA nucleotides. Smaller pieces of DNA will obviously move more rapidly than larger fragments, which will have a difficult time moving through the gel matrix. And then when the gel run is complete, the location of your DNA samples can be compared to a series of fragments or bands of known sizes called a DNA ladder. And then the presence of your fragment of interest can then be confirmed based on its size, which is determined by comparing the relative location of your test sample to the fragments on the ladder. So this is essentially what that looks like, where the fragments will move based on their different sizes, and that can then be used to provide us with the information that we need. Something that I'm just gonna give you a brief description of, because we are running out of time here. Polymerase chain reaction, PCR testing, really interesting stuff. It's actually something that I'm myself am personally interested in doing quite a bit of work with. So PCR is essentially a process by which specific portions of a sample DNA can be amplified almost indefinitely. The process mimics the biological process of DNA replication, but confines it to a specific DNA sequence of interest. So you are only multiplying the DNA that you are looking for. With the invention of the PCR technique, DNA profiling took huge strides forward in both discriminating power and the ability to recover information from very small or degraded starting samples. And then RNA sequencing. So again, this typically relies on something like the 16S ribosomal rRNA or rRNA sequence, which is essentially a fast, inexpensive profiling technique based on variation in that 16S rRNA gene. So just recall here that the 16S rRNA gene has a highly conserved and highly variable region, the conserved component of the transcriptional machinery is very suited as a target gene for sequencing DNA in samples that can contain thousands of different species. Universal PCR primers can be designed to target the conserved region, making it possible to amplify the gene in a wide range of different microorganisms from a single sample. And then conveniently, again, because of the variable region, the conserved region will make it you will make universal amplification possible, whereas the variable region will allow discrimination between specific different microorganisms such as bacteria, archaea, and microbial eukarya. So typically the workflow would be you would take your sample, you would extract it, do your sample prep, run it through your sequencer, and then look at the bioinformatics to draw you results such as this. This gives you a breakdown of the RNA sequencing and what was found as your key. If I'm not mistaken, this was for I don't want to guess. But yeah, so this is I think it was a soil, yeah, with the crust, below crust. I think this was soil samples, if I'm not mistaken. But don't quote me on that. So, and then it gives you a breakdown of what organisms are present in your samples, looking at, entire communities of bacteria or microbes. Okay, so moving on to the review questions for today. So first of all, true or false? In phylogeny, information about relatedness is portrayed by a pattern of branching, not by the order of taxa at the tips of the tree. I'm going to give you guys a few seconds if you need more time. Please feel free to pause. The answer here, of course, is true. Keep in mind here, remember back to how the tips are essentially your starting points and you're moving away from them. So they are not the ones that are telling you the relatedness, the branching pattern, where you find that universal common ancestor. That's what's telling you how related the microbes are to each other. So question 2, true or false? Phenotype is the term used in genetics for the composite observable characteristics or traits of an organism. I'm going to give you guys about 5 seconds for this one. If you need more time, again, just pause the video. The answer here, of course, is also true. Remember, something that you can use to remember this, remember phenotype and physical, they sound similar. So your phenotype is your physical characteristics. It is what you are physically able to see on the organism, or its biochemical, in the microbe sense, because you can't really see the organism. Well, you can with a microscope, and then also its results, using different biochemical tests. They're also part of this, just as a note. Then symbiosis is the interaction between two different organisms living in close physical association that is to the advantage of at least one of them. I'm going to give you about 5 seconds here. Okay, that'll be enough time. So I included this question because this can be something that might catch you guys out in some of the exams and tests. I really want you to pay attention to details here, because at first glance, this actually seems true. There has the advantage, but it is not true because of the technicality here at the end. This portion of the sentence that reads, that is typically to the advantage of at least one of them. That's not true. Symbiosis is typically advantageous to both organisms. So don't let little things like that catch you out. It's really important that you guys pay attention to detail. As a science course and as science students, many of you, and people going to the medical field, paying attention to little details like that's really, really important. Okay, let's finish off with a multiple choice question. What are the mechanisms that's horizontal gene transfer in prokaryotes? That's transformation, transduction, conjugation, C, D, and E, or E, none of the above. Oh man, sorry, that should be B, C, and D. So my answer here, sorry, I just realized that little typo. That should be B, C, and D, guys, B, C, and D. No, I'm lying. It should be A, B, and C. Wow, A, B, and C. So just ignore this question. I'm just going to give you guys the answer here. The answer here is that it's transformation, transduction, and conjugation. Those are our mechanisms. So I don't know where that came from. That was a horrible typo. So the answer here is the horizontal gene transfer mechanisms are transformation, transduction, and conjugation. Okay, guys, that'll do it for today. I will see you guys in the next lecture. Okay, guys. Cheers.